So what I would like to talk about today is innovation in patient-centric clinical trials and looking at it from a laboratory perspective. I think it's fair to say that as an industry, we're not particularly innovative when it comes to clinical trials and drug development. I think we are fairly conservative and we're not really embracing some of the technologies that we see in other industries in our clinical development, clinical trials, patient recruitment. So what I'd like to do is, as I said, focus on the patient experience. We heard from our first speaker this morning the importance of site selection, feasibility, patient recruitment, but it's also important really from a lab perspective that if we can enhance the patient experience at site, build a relationship with site, then we can have an effect, an impact on the cost of your particular study. We can use some of the web-based tools out there in terms of lab data, lab kit supplies and things like that that are, as I said, not particularly innovative, but I think that these are things that we should really try and embrace in our clinical trials. We're all under pressure in these times to do it smarter, faster and cheaper. And I think we really need to look at innovations that we can enhance to improve our clinical trials. Now, there are obvious aspects of a clinical trial where we see costs that are incurred in the trial, poor training of sites, poor feasibility, repeat laboratory testing. We've got the cost associated with getting a patient back for another visit to get another blood draw because we're missing biomarker sample data. We saw from Andrea's presentation this morning that tests are becoming more and more complicated, and you've got a lot of pre-analytical methods before you ship the sample to a central laboratory. But some of the hidden costs are in terms of recruitment delays. I've got a test case where innovation can actually help in patient recruitment and allocation of the patient onto the trial. And also, approximately 80% of or up to 80% of your clinical data is derived from the laboratory. So if you're getting errors creeping in at that sort of stage, then it's important to try and mitigate these risks early on in the process, which means that then when you come to database lock or interim analysis, if you've got good clinical data on an ongoing basis, then you can reduce time to database lock. So what we're trying to look at here is to mitigate some of the errors associated at the beginning of the process, prevention before we start on course for the clinical trial. So building the relationship with sites and supporting from a lab perspective, we can actually reduce that escalation of costs over time. So what we're going to do is look at, again, at enhancing the patient experience. As I said, we saw from our first speaker this morning the importance of site performance and enhancing the patient experience. If we can enhance the patient experience from a lab perspective, then it has a huge impact on the conduct of the study. Now, the central lab model is not new. It's been around for over 25 years, and those of you in the room who know me know that I've been involved in that sort of area for over 25 years as well. I started when I was about 5 or 6 years old, so it was that sort of approach as well. But it's not new. The kits and supplies provided to sites, the sites follow a procedure based on a lab manual, often a paper lab manual, and sometimes, as we saw from Andrea's presentation, that the complexity of the testing is becoming greater as studies become more involved. Then we've got the data that's integrated into the after analysis, and the reports are sent back to site, and your medical monitors have got the access to the results in terms of safety data. And this is important really for no matter what type of trial you're doing, whether it's a difficult niche type of unmet need in terms of a few number of patients in in the patient population, or whether it's a global clinical trial for oncology, it's important really to have access, pretty much real-time access to the data from a medical monitor in terms of safety and patient management. So really, towards 2020, innovation is really the key part of what we should be looking at. So again, looking at putting the patient first, and a couple of, as I say, innovations, although I'm not convinced that they're particularly innovative, is looking at advice that we can give in terms of, from a lab perspective, in test selection. We can look at reflex testing to try and minimize the volume of blood that you're taking, optimize the kit design and volume of blood that you're taking, and actually trying to get some more innovative ways of training the sites to deal with the patient as they're being enrolled on trial. And we've seen the increase in the number of predictive biomarkers that are coming on board now as well, and the importance of on-time results, so pretty much real-time results. As soon as your sample is assayed, your results are posted there, then you've got access to that sort of information. And I've got an example really of a clinical trial where the screening window was quite narrow for that particular disease indication. So what we're looking at is to try and use some of these techniques and technologies to maximize your budget. And one that we've seen is the use of Google Glass. It's where you can actually perform a video using Google Glass technology, and that video is delivered online to site, or it can actually be put on a CD and put as part of your lab manual. And this meant that you were able to reduce the repeat tests and false positives. And this was based on using the samples for the quantiferon TB gold assay, which requires a fairly complicated pre-analytical procedure at site before the samples are shipped. So it was cost-effective and simple to implement, and you can add translations to it if you require. And the feedback from the sponsor company and the site has been very positive. So they can see that using simple Google Glass technology to take a video of a procedure and then that get accessed by a link from site. The other thing is that most of the central labs nowadays have web-based portals, and these can be linked either whether you're with a full-service CRO or whether it's a standalone central lab. And basically, it's a portal where you can look at managing your supplies and kits. If, for example, you don't have the sufficient kits on site when a patient is coming in for a visit, then that means that the patient may have to come back at another time to have that sample taken as part of screening or whatever. And this is really your Amazon.com for ordering kits. So you can actually track and manage kits rather than have a whole cupboard full of blood sampling kits for the clinical trials that you're performing at site. And basically, you can have the web access and test verification directly at the point of sampling. So you've got access and you've got visibility on your clinical trial where you are. And often the lab data or the lab performance is a good indication of how your recruitment rates are and indication of study conduct, how well the sites are performing, rather than your traditional CRO type monitoring. So you've got access to that data pretty much real time, patient recruitment, what patient is at which visit, and so on. And also you can look at data cleaning on a real time basis. So that again, that can shorten your time to database lock at the end of a trial because you've got clean data as you're progressing through your study. So, as I said, the benefits of having the web-based portals, whether it's part of your clinical CRO type activity, that you can reduce the number of patient repeat visits, and you can pre-alert a notification of inbound samples from a lab perspective. And it's combined with the CRF data, and you can reduce your query rates to have faster, cleaner data and cut down your kit wastage. A lot of the central labs will factor in an overage in terms of percentage of kits for kit wastage. We've seen with patient recruitment where a site is thinking that they're going to enroll 20 patients in 3 months, and they don't meet that sort of target, and you've got expiration problems with kits. So if you can actually reduce your kit wastage, then you're taking a significant chunk of pass-through costs from your study trial. So what I'm going to talk now briefly about is benefits of using smarter lab data. And this was using digital pathology, and it was involved in a site. China was involved as one of the countries in the clinical trial due to the number of patients that they were wanting to enroll from that geographical area. So the central lab provides the materials for the biopsy sampling. They're transported to a local lab where the local lab prepares the slides and from the blocks. Then the paraffin blocks, stained or unstained, were sent to a local lab in China where they were prepared. Images were scanned in China because of the issues associated with getting biological material out of China, which you can't do. The images were securely transmitted to a central pathology lab, and they were 2 blinded reads. So you've got the results then back to the site. Now that is something that you couldn't do without having the digital pathology capability. So, as I said, China was a key area for enrolment in this particular trial, and it did have high patient numbers. The slides were read centrally in the US and are peer-reviewed by hepatic experts. And this, as I say, was only possible using digital pathology, which meant that enrolment in your study was back on track, and there were significant cost savings at the end of the day because your recruitment time wasn't extended and throughout the duration of the trial. Another one was, as well, is the integration of types of biomarker testing, where you've got 2 different effective types of testing, but the information needs to be combined in order to get the patient randomized or put on trial. And this was for a novel leukemia therapy where there was a tight window of the status of the disease for that drug to be administered to that patient. So what they had to do was get flow cytometry data, IHC data, combined together so that there was a central read of both parts of the data so that you could get the patient enrolled in the trial. Because, as I said, of the narrow window of treatment period for that particular drug. So again, it's taking, you may have a central laboratory that's got a great expertise in flow cytometry, but it's not got the expertise in IHC type testing. So therefore, you may need to use effectively two of your specialized labs. Some labs obviously have got the capability to do both, so therefore it's easier to combine that data and do the blinded reads. So as I say, the combination of scientific skills, we've seen, as I said from Andrea's presentation, that the trials are becoming more and more complex with regard to some of the lab parameters that you're looking at in terms of efficacy. And again, the key metrics here is it's faster, more targeted enrollment, and you're reducing the duration of your clinical trial. So in summary, really, I mean, I think what we are seeing is if with effective site training, we can reduce the pre-analytical variability of the sample preparation before they ship for analysis, and also the smarter biomarker data for patient stratification. More and more often we're seeing that the labs are becoming involved in your protocol design. So rather than cut and paste your US North American trial to a protocol for Europe, you're having more input from the lab guys to say, well, here is a biomarker that will show you some areas of efficacy within your clinical trial. And again, as I say, if we can enhance the patient experience, there's so much pressure on site performance, patient enrollment, patient retention, that if we can actually enhance the patient experience from a lab perspective, then it's got a significant impact on the cost of your particular study. So in summary, what I would like to just leave you with is, if you can work with your lab providers and try and get more patient-centric approach, drive the quality up, it will make a huge difference to your clinical trial and the outcome thereof. So, thank you.
Were there any questions for Malcolm?
To me, the process you described still seems to have the sites as the gatekeeper for access to patients and patient data. And to me, patient-centric approaches are really breaking that link and having more direct access to the patients without the site. I mean, do you get involved in any studies where you have that direct patient outreach?
Not specifically from a central lab point of view. You are correct that the investigator is king, and they are the effectively gatekeeper for a lot of that as well. I guess with web-based recruitment, then you get more access to the patient population. But from a lab perspective, we don't really see that as well. But if we can get the lab involved in terms of training sites to make it better experience for the patient, then there is a better possibility that these patients won't drop out or will be retained throughout the study.
Oh, wonderful presentation, Malcolm. And I miss my ex-colleague, Paul Travis, was supposed to present, right? Yes.
Yes, that's right.
Yes. So, and I can actually add to that. Actually, it's adding more value even to the investigators or physicians or even oncologists, because at the end of the day, even physicians are overwhelmed with the amount of biomarkers that we are actually adding more and more for prospective enrollment. And that too, as sites are becoming global, right? It's more multicultural, you're dealing with multicultural aspects. It's actually having that interactive, personalized tool via these web-based portal systems wherein physicians can actually educate the patients who are already under trauma. And because already they're going through grueling diseases, so they can understand what exactly is going at the level of the lab. Because already the local lab has diagnosed them as stage 2 or stage 3. But as per the regulatory requirements, you need a lot of central lab diagnosis as well. So like patients are always under pressure, like when will I be enrolled into the trial? So these tools have been really like taking the patients through through a peaceful journey, I must say, in that context, wherein I could be enrolled, and they're actually doing some innovative tests to better diagnose and also to better enroll into a right scenario wherein the right patient is getting the right drug via the right test.
Yanis Karayouros, BMS, thank you very much for actually sharing a few examples of where the central lab acts as an integrator of specialty coming sometimes from subcontractors. What we see is a growing level of awareness and experience by central labs in integrating successfully and operationalizing this type of data. However, it becomes increasingly difficult to keep up with sometimes conflicting regulatory expectations, in particular between the US and Europe, as to the auditability of the data. It's fine to have it done on time. We probably are very good in tracking timing deviations or protocol deviations. But when you go back a couple of years later and wish to reconstitute the trail of events, in particular associated with endpoints, we seem to be getting a lot of scrutiny. And at that point, it's the sponsor company that gets the scrutiny because the audit trails are no longer available. What is your position? Do you proactively try to record things in a way so that they are auditable in the longer run, or you are transparent to your sponsors and you tell them, you know what, make sure that if you have such requirements, put an agreement in place for us to be able to retrieve this data and transfer it to you at the end of the trial?
I think, thanks for the question. I think from a regulatory point of view, I mean, we've got to, as a laboratory, be able to have that audit trail all the way through. So when the sample was arrived at the facility, when it was testing, what the tests were, and what the QCs associated with that batch of sample analysis. So we should really have the data, or the lab should have the data available for an inspection. They all adhere to the various guidelines. If you're doing bioanalytical in terms of PK data, then it's a GLP compliant laboratory that would be doing that test. So therefore you would have the audits in place. I'm surprised a little bit to hear that, you know, 2-3 years down the line you're audited and then there's been an issue with the data that has been generated by a clinical trial, because it is date and time stamped, whether that information, the results are going back to site, or whether it's actually part of your database submission.
Sorry, to clarify my question, the difficulty starts because of much longer term expectations. Okay, everybody knows about pediatric studies, we have to keep the data forever. But it seems, I don't know, we have been talking about the directive a lot, we have kind of forgotten all these days the E6 upcoming amendment. And the new version of E6 will actually expect data to be stored. So that's
right. Okay.
The data is saved for 25 years in US, right? So, okay. Not in Europe? Wow, okay.
Hi, Marco. We're seeing a lot of unexpected, maybe, or not unexpected new players in the healthcare market that are now also trying to enter the clinical research market. I think you already mentioned Google, the Apple Health. How do you see their roles in this whole patient experience and central labs with so much now happening more at home rather than in the clinic?
I think that's a good point. I mean, I think we shouldn't be afraid of some of the technologies that we're seeing, as I say, from Google Glass and Apple and what they're getting involved with. And we also see a lot of point-of-care testing, particularly, and you've got studies, oncology trials, where the patient safety and for dosing or treating that patient is important. You've got to get that safety information quickly. But you're still having to go back to the traditional, if you like, a central lab type of testing, where you've got consistency of results as part of your efficacy when your study's unblinded at the end of the trial. But yes, I mean, we should embrace the new technologies that are from the new players in that sort of area. But I guess what I said at the beginning is that as a pharmaceutical industry, drug development is a very conservative process, I think, and the changes taking time to be implemented. These things that I was talking about there, they're not particularly innovative, really.
Thank you.